Hello guys, this is Dr. Komal, CTVS surgeon. Our very own website CardioBeat was started with an aim of helping students who are doing residency in cardiac surgery and cardiac anesthesia by providing blocks, diagrams, notes, PPT presentation as well as video presentations. So as a part of this initiative, we would like to present a video presentation on development of heart. So the embryology of heart will be discussed under the following subtopics, namely introduction, establishment of heart field, formation of heart tube, cardiac looping, sinus venosus development, development of venous system, arterial system, conducting system, and septation of cardiac chambers. For the ease of understanding, the embryology of heart videos will be divided into three parts, and the first part we will have an understanding of introduction, establishment of heart field, formation of heart tube, and cardiac looping. So let us quickly move on to our first subtopic, which is introduction. A basic understanding of development of embryo is very much necessary for understanding development of heart. So as we all know, the oocyte and spermatocyte, they combine to form zygote, which is a one cell or a single cell. And this subsequently undergoes multiple divisions, forming a 16-cell stage called as morula. Initially, the morula is a clump of cells, but later segregate itself into an outer cell mass as well as an inner cell mass. So, this outer cell mass is called as trophoblast, whereas the inner cell mass is called as embryoblast. This outer cell mass, trophoblast, divides into syncytiotropoblast and cytotrophoblast, which later on subsequently gets attached to the maternal endometrium, forming the placenta. Our focus of study is on the inner cell mass, otherwise called as embryoblast. Now, we will quickly move on to the changes that is happening during the second week of intrauterine life. So, this embryoblast, which is a mass of cell at one pole of the embryo, starts segregating itself into two layers of cells. We have the dorsal or the external layer of cell and a ventral or internal layer of cell. So this mass of cell, embryoblast, so this is the embryoblast, which is mass of cell, gets segregated into two layers. So we will have these two layers in different, we will draw these two layers in different colors. So we will take this external layer into blue color and the internal layer, we will depict it with the yellow color. So this external layer has a special name. So the special name of external layer is called as epiblast. An internal layer has a special name called as hypoblast. So this mass of cell now has two layers, epiblast and hypoblast. Okay, so this epiblast starts giving newer cells, and this newer cells will start migrate away from the epiblast, forming a ring around the epiblast. Similarly, the hypoblast is going to secrete newer cells which starts migrating away from the hypoblast as well. So thereby this embryoblast, now which is segregated into dorsal epiblast and ventral hypoblast, has already secreted newer cells which is moving away. Thereby this embryoblast has now contains two coelom or cavity. So the cavity which is lining inside the epiblast is called as amniotic cavity, and that which is lined by the hypoblast is called as the primitive yolk sac. So we'll move on to the bigger picture. This picture, we will put this embryoblast into that picture and see what is happening. So we have the outer trophoblast. Then we have what is called as the epiblast with its own cavity, which is the amniotic cavity, and we have the ventral hypoblast, which has its own cavity, the primitive yolk sac. So when these two have their own cavities, namely the amniotic cavity and a primitive yolk sac, they should be also an cavity which is lined by the trophoblast, which is this cavity, which is called as the chorionic cavity. Well, hope you understand. So we have started at the second week of intrauterine life, where the embryoblast is segregating with its own layer of cells and own cavities. Thereby, the second week of intrauterine life, the embryoblast has got two discs, which is the embryoblast has two discs, which is the hypo and the epiblast. So this is otherwise called as bilaminar embryonic disc. So at the second week of intrauterine life, the changes that are happening in the developing embryo is going to result in the formation of the bilaminar embryonic disc. Okay, let's move further. So we will move quickly to the third week of intrauterine life. So the changes which are happening during the third week of intrauterine life. For this, we will take this part of embryoblast. Okay, so this part of embryoblast. So we have the hypoblast, epiblast with their own cavity, yolk sac and amniotic cavity. So I'm just, for the purpose, will just take this part of this embryoblast and we'll start drawing it in a larger section. So this is the epiblast and this is the hypoblast. Right. So the embryo is going to have its own cranial end as well as the caudal end of embryo. So at the third week of intrauterine life, a streak of cell forms at the caudal end of the embryo, and the streak of cells start adding newer cells, which starts traversing between the epiblast and hypoblast. So initially the two layers were closer to each other with no separation. They are attached to each other. But because of the addition of newer cells formed from the streak of cells, they are getting separated at the midline. So this streak of cell is called as primitive streak. And the newer cell which is traversing cranially between these two layers have a node-like structure at the cranial end called as primitive node. As more and more cells are traversing, more and more cells are forming cranially to the primitive node. They form a cord-like structure called as notochord. At later part, due to the signaling pathways, this primitive streak notochord, they undergo what is called the formation of neural plate, which subsequently forms neural fold and grooves to form the future spinal cord and brain. So, one thing that is happening, the change that is happening during the third week of intrauterine life is that formation of primitive streak, which adds newer cells which are going to traverse between this epiblast and hypoblast. So they get separated in the middle. So I would like to draw another section of embryo so that we'll be able to understand better. So this one section of embryo where you have the epiblast as well as the hypoblast. Sorry, this is in yellow color. And in the middle, they are separated by the primitive streak notochord. Not only this, the cells are not only traversing the epiblast and hypoblast just at the middle or the center, but the cells are being flowing laterally. So that we will depict with a green color. So not only they are traversing in midline, so this is a cross section of embryo at the midline, but they are also traversing laterally. And they're forming a sheath of cells laterally separating the epiblast as well as the hypoblast. So this sheath of cells is what is called as intraembryonic mesoderm. So epiblast, hypoblast are now separated in the middle by means of intraembryonic mesoderm, which is nothing but a group of or sheath of cells at the either lateral end of the primitive streak and notochord. So at this junction, I would like to modify the name of epiblast and hypoblast as future ectoderm, and hypoblast is becoming future endoderm. This intraembryonic mesoderm is further getting segregated into three from its medial end to the lateral end. So how we are going to draw that? Let me show. So the medial most part of the intraembryonic mesoderm, this is the most medial part of the intraembryonic mesoderm. And then we have a middle part of intraembryonic mesoderm. Let me take another color. So this is the middle part of this mesoderm. and extreme lateral part. So this intraembryonic mesoderm itself is divided into the medial part, middle part, and the lateral part. So the most medial part is called as paraxial mesoderm, and the middle part is called as the intermediate mesoderm, and the most lateral part is what is called as lateral plate mesoderm. So in future, paraxial mesoderm is going to give rise to the axial skeleton, the somites. Intermediate mesoderm is going to give rise to the genitourinary system, and the lateral plate mesoderm is going to give rise to your heart. So with this, we have just completed the introduction part. Sorry, I forgot to tell you about one thing. So this division of cells into ectoderm, mesoderm, and endoderm, which is happening during the third week of intrauterine life, is what is called as formation of trilaminar embryonic disc, otherwise a process which is named as gastrulation. And this gastrulation is very much necessary in the development of embryo, because without gastrulation you don't get any of your organs, because it is necessary for embryo to get divided into ectoderm, mesoderm, and endoderm for the formation of various vessels and what you called as the organs, the skin, the somites, the developing brain. Okay, so with that we have completed the subtopic, just a subtopic of introduction.